Hi folks, let's get into the prokaryotic envelope a little bit further, okay? We were talking about the glycocalyx, this extracellular matrix that is around the prokaryotic cell. It could be a capsule or a slime layer, or there may be no extracellular matrix associated with prokaryotes, but there are a possibility. But a lot of cells, a lot of prokaryotes will have a cell wall, with a few exceptions, okay? So as we dive deeper into the prokaryotic envelope, we run into this cell wall, okay? Generally, we would say that the cell walls of prokaryotes are made of peptidoglycan, and the word tells you a little something, right? Peptido is something to do with peptides, okay? So a peptide is a small protein, okay? A linkage of amino acids together. Again, something that you were kind of brushing up with your chemistry assignment. And then glyco or gluco sound a lot like this last part of the word, right? So this is a protein and a protein and a sugar coming together to make this wall, okay? So peptidoglycan layers to make up these prokaryotic walls. Now the carbohydrates make up the backbones, and the polypeptide cross bridges make up the peptide part of that, okay? So let's switch to red here just because I think we'll see it a little easier. So if we look at the carbohydrate backbones, we run into something called N-acetylglucosamine, or we'll just say NAG, okay? And you see the gluco in there, so we know it's a sugar, something or other, right? And then we have acetyl muramic acid, which we'll just say as NAM, okay? So these two together make up the carbohydrate backbone, okay? So we have these backbones, the NAGs and the NAMs, okay, coming together to make up the backbones of this cell wall. Now the polypeptide cross bridges, okay, so we'll switch back to green for this. They are going to be made primarily of glycines, okay? So glycines are going to tie together these, and you see in green here the lines I'm making, right? They're going to tie together these carbohydrate backbones, okay? And I always think of this almost like a log raft, right? So you have these peptide linkages that are going across, right? Like these ropes going across the logs, right? And then we have these logs that actually make up the NAG and the NAM parts of that carbohydrate backbone, okay? So this all ties it together. Now, the functions of the cell wall are numerous. One of them is that it prevents osmotic lysis. So, you know, if we talk about animalian cells, there are water pores in animalian cells, in the membranes of those cells. Of course, animalian cells don't have a wall, you remember. And water is able to move freely in and out of these cells. Now, in this case, though, a wall provides stability to the outside edges of a prokaryotic cell. So if a prokaryotic cell is sitting in water, well, water is pure on the outside. There's a concentrated mix of things in the water on the inside of the cell. So where water is more pure, it will move from high to low, right? So we're going to have water going into that cell, but it will not pop the cell like it would an animalian cell, right? Also, this regulates the movement of ions, you know, minerals and electrolytes in and out of the cell. So it plays a role in what gets in and out, okay? Now, if we were to look at the difference in two major groups of prokaryotic cells, we would see that there's a big difference in two major groups in that one has a very thick peptidoglycan layer in its wall, and others have a very thin peptidoglycan layer, okay? So again, if we go with this red sugar backbone, right, carbohydrate backbone, and then we'll put in green again these peptide layers that are going across, right, these glycines. We see that again, right? Well, in an organism that is considered gram positive, it has a thick set of layers of these peptidoglycan rafts. So think about one log raft on top of another on top of another, okay? So here, let's switch to a color. We'll go with orange here. So teichoic acids are known to tie together. So we see these layers being tied together by something called teichoic acid. Okay? So gram-positive organisms have many layers of this peptidoglycan, thick peptidoglycan walls, and teichoic acids are known to be associated with those gram-positive organisms. Okay? If we look at gram-negative organisms, they have just one log raft. Okay? It still has the carbohydrate backbone, right? You can see that here. And it's not showing this, but you still have glycine that would actually tie these together. But you will not have the teichoic acid, okay? Because we don't have layer upon layer of peptidoglycan, okay? The other thing that we see with a thin gram-negative wall is that we see an outside outer membrane outside of the wall. So we have this inner membrane, which is the plasma membrane, which of course we see here with a gram-positive wall, just inside the gram-positive wall as well. But we have this outer membrane in the gram-negative cell, which provides us with this gap in between two membranes. This gap in between two membranes is known as a periplasm. Okay, so here they're talking about this periplasm that exists between the two membranes in a gram-negative. We don't have that little gap in a gram-positive cell. Okay. So what are some of the characteristics of a gram-negative outer membrane? This outer membrane that's not existing in the gram-positive cell. This protective coat has lipopolysaccharides. It has lipoproteins. So we say lipo, it's something to do with lipids, right? So fats. So we have these fatty kind of sugar molecules, fatty kind of protein molecules, and of course phospholipids that we know exist in every membrane anyway, right? We have this bilayer of phospholipids. But one big thing that I want to really bring to your attention here is this lipopolysaccharide, okay? Lipopolysaccharides are known to be associated with the outside membrane. This outer membrane of lipopolysaccharides is really important to understand and realize they exist when it comes to treating an infection that is caused by gram-negative organisms. These lipopolysaccharides can be a real problem if they are allowed to disperse within the body or within the bloodstream, let's say. Okay? The immune system reacts in a resounding way to this lipopolysaccharide, and they often say L, P, and the S, so we say LPS molecules when we talk about these. Okay. Just to give you an idea, when we are treating infections that are caused by gram-negative organisms, we don't want to apply antibiotics that destroy the cell wall, because what this does is it disrupts these polysaccharide molecules and disperses them in the bloodstream. Now, the immune system sees these LPS molecules as an abundance of foreign molecules. And oftentimes this will cause the body to react with a huge inflammatory response. Okay? So this results in anaphylaxis. It results in a really negative effect ultimately. Okay? So we want to pick and choose wisely based on whether the infection is caused by gram-positive organisms or whether it's caused by gram-negative organisms. Now, something also important about the outside membrane here is that it actually does affect how phagocytes interact with these organisms, how it is that antibiotics interact with these organisms, like I already mentioned. Also, within the periplasm that I already mentioned with you guys, right? So we have this periplasm that's a kind of a little gap here in between the two membranes. This periplasm for gram-negative organisms will often contain protective enzymes. Remember that these bacteria exist in a nasty world where they're competing for nutrients and space and all of those things. And bacteria actually are affected by viruses as well. It's not just animals and plants that have viruses attacking them. Bacteria do too. So within this periplasm, we have protective enzymes, some of which will break down genetic material. So if a virus is about to inject its genetic material into a bacterium, the periplasm will protect it. Okay? All right, so going forward, we will talk about this section down here. Right now, when it comes to the first exam, I wouldn't worry too much about all the stuff you see that I circled in green, okay? What I would worry about more so would be, let's see here, these aspects that you see above that point, okay? So you'll want to understand some of the differences between gram-negative and gram-positive organisms when it comes to how they would stain, if they would stain purple, or whether they would stain red. Okay, I should probably change the color, right? Whether they would stain purple for gram-positive, or that whether they would stain red. Let's change the color here, right? So purple for gram-positive. So purple and P and P, right? So purple are positive, and the negative ones are going to be red. Okay. Also, you want to know about the teichoic acids, whether there's a periplasmic space, outer membrane, all these things that we already just went through. All right, so let's just run down through how a gram stain actually works, all right? Because this is something that we've already gone through when it comes to the lab section, but it's really important we have this gram stain down. So if we're staining bacteria in a lab, one thing we'd look for maybe is the cell shape and arrangement, but we can see that with a simple stain. We can see that with a stain where everything would come out the same color. But here now, with a gram stain, we're trying to differentiate between organisms. Earlier, we differentiated based on colony, size, shape, color, odor, all of those things. We can use that. But now on a microscopic level, we might want to look at these cells in the microscope and stain them, all right? So the first step is to heat fix the bacteria. Go about that process like we see in lab, where we would put the bacteria on a slide, pass it over to flame, makes those bacteria stick to the slide, and it kills the bacteria. And then we go about applying different stains. The first stain would be crystal violet. This is purple. It's going to stick to any of the peptidoglycan walls. whether it's thick or thin. Okay, so after this is applied, you're going to have purple on all cells, as long as they have a peptidoglycan wall. The next thing we would do is apply this mordant, a mordant called iodine, okay, or Graham's iodine. What Graham's iodine will do is it will actually kind of fix the crystal violet extremely to the thick walls, okay? It's not going to come free from those thick walls very easily once iodine is applied. Then we come along with a decolorizer, Graham's alcohol, which is a 95% ethyl alcohol. We apply this until the alcohol runs fairly clear off of that slide. Once that is done, we now would know that the stain has come free from the cells that don't hold the stain very well. So that purple color will stay on Gram positive cells, okay? But it's going to rinse free from any Gram negative cells that happen to be on that slide. So if we were to put the slide under the microscope at that point, we would see no color associated with those cells, those Gram negative cells. Then we come back and we apply a counter stain. This would color any of the peptidoglycan walls that are left without any color, right? Which would be those Gram negative cells. And now we would have a red or a pinkish color associated with those Gram negative cells, and the Gram positive cells would remain purple. So this is now going a step further. It's not just cell shape and arrangement we get to see. It's also a difference in color based on the cell wall structure, something structurally different about those two cells. This would be differentiating among them. All right, the last thing I'd like to mention is that some prokaryotes do not have walls at all, okay? Archaea, for example, do not have walls at all. Okay, they're wall-less. They're similar to eukaryotes in that way, and they're more related to eukaryotes, believe it or not, in that way. Okay, like our own cells. Even though archaea, remember, are these crazy cells that live in really hot, acidic places. Okay, but there are some prokaryotes, some bacteria, that also don't have walls. One example would be mycoplasma. Okay, mycoplasma, if you've ever gone, I'm sure maybe you've experienced this, if you've ever gone in or heard about someone who's gone in, they think they have bronchitis or pneumonia, and they're sent away with some antibiotics, and they don't touch it, right? So the person goes away, the signs and symptoms get worse, and they go in and they might do some further exploration and find that maybe you have a different kind of pneumonia. All right, why didn't that first set of antibiotics work? Well, what they were probably doing is taking an educated guess that the organisms causing your pneumonia had a cell wall and gave you an antibiotic that would kill those organisms with a cell wall. Well, mycoplasma has no cell wall, okay? They call this atypical, so it's not the typical pneumonia, right? So atypical or walking pneumonia, all right? So they have to put you on a different set of antibiotics, a set of antibiotics that don't target the cell wall of bacteria, but in fact target something else, something that else is critical to the survival of those cells. Okay? And we'll get into antibiotics in much more detail later in the course, in the third section of the course, in fact. But for now, let's leave it at that. And next time we're going to dive a little deeper into the cell, look at the membranes and flagella and even subcellular structures. Okay?